before Jonathan gets started, I'll just give a tiny bit of an introduction. So Jonathan Polimeni is an assistant professor of radiology at Harvard Medical School, as well as an assistant in biomedical engineering at MGH, Massachusetts General Hospital. He's part of the MRI physics and instrumentation group at the Martino Center in Boston. And he started his career actually at Johns Hopkins before going and doing a PhD in electrical engineering from Boston University in 2007 on measurements and models of visuotopic maps in macaque and human visual cortices. His work focuses on technological developments that increase the spatial resolution and accuracy of functional MRI. And he has a particular focus on understanding the biological limits and spatial specificity of cortical layer signals. Today, he's going to talk to us about the utility of biophysical models for interpreting fMRI signals. And he's going to relate those signals back to neuronal activity. So in other words, the inverse problem. So with that, I'll let Jonathan take over.
Great. All right. Thanks, Tyler. I'll try my best. So thanks for that introduction, and thanks again to Luca and Renzo and Tyler for inviting me and for organizing this dinner. And so when Luca and Renzo first proposed to meet as a group in Sydney during ICMM for pizza and talk about topics relevant to layer-fMRI, I thought this was a terrific idea to help this small community discuss new data, new analyses, and perhaps debate ideas about how to achieve our shared goal of measuring brain activity across layers. And so based on this, I wanted to take this opportunity to present one approach that our group and other groups here have been developing to try to achieve greater neural specificity from our fMRI data and extract more relevant information about activation across layers. Now, this biophysical modeling effort is a work in progress, but I wanted to share with you a snapshot of where we are and share some ideas of where we're taking this. So let's see if I can advance my slides. Okay, great. So so the premise of this work is that, as this group I think knows well, there's mounting evidence, much of which is from our friends in optical imaging, for perhaps surprisingly high precision hemodynamic regulation in the brain. This opens possibilities that distinct fine-scale patterns of neural activity can elicit distinct patterns of hemodynamics. We've seen many times now that fMRI can distinguish the site of activation across cortical depths, which can be used to infer the pattern of activation across layers. However, all fMRI contrasts are of course influenced by local vasculature. Therefore, models can aid in the interpretation and potentially to relate the fMRI measures back to the underlying neural activity. So now previous attempts to model the fMRI signal in the context of laminar fMRI did so by simplifying the vascular anatomy, and I think for good reason. This helps to provide tractable models that can be used in practice, but there are some limitations here. Now, these models generally assume a fixed number of independent capillary compartments coupled by radial intercortical vessels, either diving arterioles or ascending venules. Now, one of the most useful models is the Havlicek laminar bold response model, which is a generalization of the balloon model. This model fits the data really well, but the physiological interpretation of many of its parameters is still uncertain. And so these values have to be tuned, which limits the generalizability of these models. Also, the arteriolar dynamics, including the time evolution of dilation across the arterial tree, are often missing. So now these models shown here are incredibly useful and it taught us a lot. So our goal here is to develop more biological models with more realism to inform these simplified tractable models, which can help to determine, for example, the laminar spread functions or the model parameters with more confidence. Okay. So in summary, our goals are to generalize the current modeling framework to account for more data. We also want to use these more realistic models to gain insights into the spatiotemporal resolution limits of fMRI and to help improve our mechanistic understanding to infer neural activity from the fMRI data, or the so-called inverse problem, which I'll touch on briefly at the end of the talk. Now, towards these goals, our group has been developing biophysical models based on realistic microvascular anatomy and dynamics. This is in close collaboration with David Boas and David Kleinfeld. So we start with the full microvascular reconstructions of the mouse cortex and lung spot. We compute blood flow dynamics and oxygen transport, then calculate the associated magnetic field offsets, and finally, the corresponding bolt response dynamics. So now this approach is called the Vascular Anatomical Network, or VAN modeling framework, since it differs from other fMRI modeling approaches, such as those based on the balloon model. Now, in a bit more detail, this approach was introduced by Louis Gagnon and David Boas's group. Start with a reconstruction of macrovasculature from a spot of cortex. They started with about 600 micron cube. Now, each network is about 1 input artery, 6 output veins, and about 1,000 capillaries. So we simulate the full spatial and temporal dynamics of the BOLD response based on boundary conditions for blood pressure and oxygenation. The inputs here are time traces of arteriolar dilation or diameter changes, which represent the vascular response to neural activity. So now in the first stage of the simulation, we use fluid dynamic computations to simulate blood pressure, flow, and volume changes. In the second stage, blood oxygenation is computed using a model for tissue oxygen consumption, and the simulation of oxygen advection in the vessels, also exchange between red blood cells, plasma, and tissue, then oxygen diffusion into the tissue. Now, all of these quantities are defined at the single vessel level and are based mainly on data from invasive optical microscopy measures, as opposed to typical fMRI models that are based on MRI measures at the voxel level. Then in the final stage of the simulation, we use a water diffusion simulation to compute the BOLD response within a given set of voxels of a chosen size. So briefly, I should point out that this framework is clearly quite complicated with lots of parameters and assumptions, and therefore there's some danger of overfitting. However, many aspects have been validated against two-photon microscopy measurements in the mouse cortex. So here's an example of the computed partial pressure of oxygen simulated across the vascular network compared with optical measurements. And so we see a quantitative comparison of the simulated and experimental distributions across the vascular network is shown here, indicating a reasonable match. So validations such as this provide us with some confidence in these simulations. So how do these VAN models differ from random cylinder models, which were the classic approach for modeling vessels to understand vascular influences on the BOLD signal? So they differ in terms of both the geometry and the topology of the vessels. But by geometry, I mean that the VANs have accurate distributions of vessel diameters, orientations, and densities, all of which have a strong influence on the fMRI signals. And by topology, I mean that the connected vessels in the VAN allow for simulation of dynamics, both blood flow and oxygen transport, and the time evolution of these vascular responses. So now, while random cylinder models are helpful, and I'll show you some results later in this talk with the random cylinders, VAN models are better suited to understanding how vascular geometry and the topology shape the fMRI responses. Now, so the initial VAN modeling work of Louis Gagnon provided some new insights into the BOLD signal. For example, the BOLD dependence on cortical orientation, which could only be observed in vascular models with realistic anatomies. So now, although this orientation dependence of a single vessel is well known, Louis showed using this VAN model, quantified through simulations, this orientation effect expected from a voxel containing realistic microvascular anatomy. In other words, they calculated how the BOLD signal amplitude varied with orientation to the cortical surface normal relative to B0. So now some orientation effect is expected since the largest penile vessels run parallel to the cortical surface. But what Louis predicted was that there is a whopping 40% effect. So that is, the BOLD response should be 40% larger in cortical locations where the cortex is parallel to B0 compared to locations where it's perpendicular to B0. Now, the size of this effect was somewhat surprising, and so they went down to our 3G trio scanner and did a whole brain activation task using a CO2 delivery, and found that they could observe a similar amplitude of this orientation effect in the in vivo data as well. So this is a nice example of how this approach could make a new prediction about the BOLD signal based on realistic VAN models that could then be tested and observed in some conventional data. So with this motivation, in this short talk, I'll briskly cover 4 topics that roughly correspond to some abstracts presented at the ISMRM last year, so you can go to these abstracts for more details. First, we extended the VAN model with realistic blood pressure and vessel compliance. We're also working towards a VAN model for the human cortex. We've been performing fMRI modeling with realistic image encoding and gradients to understand how image encoding kind of affects contrast encoding. And finally, we've been doing some forward modeling using subject-specific vascular maps. And then again, at the end, I'll share a few aspirational thoughts on applying this approach to the inverse problem. Okay, so first, we sought to extend the VAN framework to account for missing supplying and draining vessels. So this is work from Jörg Vanmoller from my lab. So one important aspect of this simulation is that we use a small VAN representing a single location in the cortex. But to generalize the responses that match what we measure in vivo, we have to account for the anatomical context, or all of the supplying vessels, mostly arteries, that are missing from the VAN that supply and drain this one spot of cortex. Now, this can be addressed by adapting the boundary conditions to account for the missing upstream feeding arteries. So I'm not going to go into too much detail here, and we presented this last year, but it's an important aspect of this modeling work since the pressure delivered to the vessels has a major impact on the hemodynamics. So as an example of how this modeling framework can provide some insights, in this study, we wanted to examine whether capillary dilation may cause measurable changes in the bold response. That's because today it's still a bit contentious whether capillaries do dilate in vivo, whether actively or passively. So here we use the model to try to toggle the vascular compliance of the capillaries. The solid lines here correspond to the simulations with compliant capillaries that were allowed to dilate, and the dashed lines correspond to simulations with non-compliant capillaries that did not dilate. We also examined the bold response separately in the capillary and venous compartments to get a sense for to what extent the total bold response was similar to this capillary response. Now, perhaps unsurprisingly, the passive capillary compliance essentially extends the venous balloon to the capillary bed, which causes an increased dynamic decoupling of CBF and CBV that manifests as a more pronounced bold poststimulus undershoot when the capillaries are compliant. Now, while we think that these responses are actually a little bit exaggerated, and we're working towards making the simulation more realistic, this simple example shows how the VAN framework provides a little laboratory to test out possible scenarios of neurovascular coupling and associated hemodynamics. So next, we wanted to investigate a prominent feature of the bold response that provides a lot of insight into the various hemodynamic components, which is the bold poststimulus undershoot. Now, this is classically attributed to a delayed cerebral blood volume recovery to baseline, and it's one possible cause of the bold undershoot. Now, for short duration stimuli, less than about 4 seconds, this delayed volume appears to be reduced. And so we wanted to get some insights into whether this might be this appearance of a bold poststimulus undershoot for longer duration stimuli may have some sort of mechanical or anatomical origin. So we tested whether this framework could help us to account for this nonlinearity of the hemodynamic response to short duration stimuli. Now, it's known that pronounced bold poststimulus undershoot is expected for short duration stimuli. And indeed, the balloon model can capture this phenomenon only when using different parameter values as shown here. For responses to a short duration stimulus in blue on the left and a long duration stimulus in red. And we presented this year how the VAN model simulations for both short and long duration stimuli using a fixed set of parameters can capture these expected effects. We do observe a pronounced bold poststimulus undershoot only for the long duration stimuli. Therefore, the VAN model in this case appears to generalize well, and with a single set of biophysical parameters, it can account for this observed and well-known kind of nonlinearity in the bold response. Now, we've revisited these simulations, and now we think that the pressure delivered to the veins here is too high. It may only be appropriate if most of the brain were activated, and so we're currently repeating this experiment with more realistic pressures, which I think was going to reduce the amplitude of this undershoot. Also, the amount of cortex that's activated may have an influence on the blood pressure that's delivered to the active cortex, which could also affect these responses. So we're working on this now. So we wanted to test whether the VAN modeling framework could account for the varying bold response features across depths. It has been shown both in animal data on the left and human data shown on the right that the hemodynamic response shape varies with cortical depth. In particular, the post-stimulus undershoot is more pronounced to the PO surface, but the reasons for this are still a bit unknown. And so to understand this phenomenon, we were inspired by a recent study that reported different biomechanics of intracortical and PL vessels. They reported that the compliance of the parenchymal blood vessels was lower because they were surrounded by tissue, which constrains their dilation, whereas vessels on the cortical surface were less constrained and therefore more compliant. So they observed this by observing the dilations of parenchymal vessels surrounded by tissue and PL vessels after controlling for vessel size, branching order, and other effects, and observed this both in arteries and veins. So based on this, we extended our VAN simulations and assigned viscous and elastic properties to the individual vessels in the VAN based on whether they were surrounded by tissue or not, and using tissue and vessel elasticity and viscosity values from the literature. Note that this is one major difference between the VAN models and more common lumped models here. The microscopic parameters are set for each individual vessel instead of setting macroscopic or lumped parameters for the entire voxel. And so with these material properties set, we're then able to estimate the pressure and volume responses in the VAN to the input flow response. So we also presented at ISMRM that we found that after assigning these elasticity and viscosity parameters to the vessels and rerunning the VAN simulations, we indeed saw that the simulated PO vessels were highly viscous, exhibited a slow return to baseline, and exhibited this pronounced bold poststimulus undershoot compared to the responses that you could see in the parenchyma. Now, again, these poststimulus undershoots are, again, a bit exaggerated due to the large pressure that we delivered to the veins here, and we're now recomputing these results and expect a smaller effect. So that's a work in progress. So there are many limitations of these models, of course, but are they useful? So I think we do see some value added over the balloon model in terms of generalizability. Now, future work will be used to use this framework to update or inform the balloon model, for example, by passing parameter values to Camille and Martin to include in their laminar bold model, which is much easier to use in practice. So we're looking for a model that's as simple as possible, and the VAN framework can potentially help determine balloon model parameter values. Fortunately, I should say, the model parameters are interpretable based on tissue material properties such as vessel wall elasticity and viscosity, which can be measured separately. Of course, there's a risk of overfitting and more validation data are needed. So one challenge that we faced was how to set these boundary conditions, the pressure boundary conditions at the border of the van. And so we sought to generate larger van models, which can potentially provide more accurate flow and pressure distributions. So to create larger van models, we started by trying to reconstruct larger vans from our microscopy data by segmenting the vessels. But since even small segmentation errors can cause large problems with the topology, after some discussions with Natalia Petridou, we decided to pivot towards synthesizing vans with similar anatomy and topology to real vans. So here you can see an example of a reconstructed van and 6 statistically matched synthetic vans. This is work by Grant Hartung, who recently joined the lab. So after comparing the flow and oxygen dynamics, Grant was then able to simulate bold responses from these reconstructed and synthesized vans, showing that the two were reasonably similar. So this gives us some confidence in the validity of these synthetic vans. So we can also synthesize vans with parametrically varying angioarchitectures, such as capillary density, which can help us to evaluate the influence of non-uniform CBV across depths on the bold signal. This also demonstrates how we can potentially generate van models for different brain regions given some prior knowledge of the vascular density or architecture. We are now working towards synthesizing and simulating a human-sized van model. Here you can see a candidate van for the human cortex next to a van from the mouse cortex to give you a sense for the differences in sizes. Oh, sorry. So after dramatically improving the computational efficiency, oops, sorry, I was just answering. Okay, we dramatically improved the computational efficiency of our flow and oxygen solver, and we're now able to compute oxygenation in this human van in a reasonable amount of time, although still a full simulation takes a few days to compute. So what are the main challenges with the van modeling framework? Well, first of all, this framework is computationally demanding, with a single 40-second bold response requiring something on the order of hours to days to compute. Also, there's a large number of physical parameters that are obtained mostly from small animals, and so the question is, how generalizable are they to humans? We also need to account for subject-specific vascular anatomy. We're not really sure how consistent the vascular architecture is across individuals. For these simulations, we're making many assumptions about the vascular response. We're only using available dilation traces measured from one stimulus condition, so we're thinking about trying to model this sort of coordinated vascular response. We also have to make many assumptions about oxygen consumption and neural activity. Now, these first 3 challenges we are addressing now, but tackling all of these will require close collaborations with friends performing invasive measures of these dynamics. So we've also been looking at trying to understand the effects of image encoding on the fMRI signal using similar modeling approaches. Previous simulations, such as the classic Boxman plot here on the left, have shown that for pure spin echo or T2 bold, we expect a reduced signal change around large vessels with large radii, as seen in this bell-shaped curve here. Now, with spin echo EPI, it's difficult to achieve this microvascular specificity in practice. Now, I'm sure many of you are familiar with this study by Bunze and Logothetis, but as a reminder, here they showed that the spin echo EPI with typical readouts contains a lot of T2 star contamination, and the measurements show strong influence of large veins on the PO surface, as shown here in this top panel. But as they reduce the echo train length by segmenting an EPI readout, they're able to gradually achieve a microvascular specificity expected from a pure T2 bold acquisition, which you can see here in the bottom panel. And so because of these effects of image encoding, we wanted to incorporate this into our modeling. And so we first set out to see whether we could reproduce this effect of spin echo EPI readout duration on the microvascular specificity. And so Avery Berman from my group has started to incorporate the details of image encoding process in our simulations, currently based on simpler random cylinder models to allow us to more easily sweep the vessel radii to model the microvascular specificity. of spin echo EPI. For this, we literally impose image encoding gradients in our simulated voxels for various acquisition times, or TACQ, as defined here, to better understand the role of the EPI acquisition on bold simulations and the unwanted T2-star contamination in conventional spin echo EPI. So here is a Boxman plot of the sensitivity of spin echo EPI bold signal to various vessel radii. We first see that for an idealized spin echo EPI with an infinitely short acquisition time of 0 milliseconds, we observe the expected microvascular specificity. We're essentially reproducing the classic results of Boxerman. But as we simulate longer acquisition times in the spin echo EPI acquisition, we see that while there's some increased variability, there is an overall increase in the spin echo EPI bold signal for larger vessel radii, as expected from the empirical data of Gunza and Locathetis. So then, in order to understand the cortical depth dependence of this macrovascular contamination, we built a simple vascular configuration consisting of a large PL vein on the cortical surface sitting on top of a model cortex consisting only of a microvascular network. We then simulated the spin echo bold signal for voxels at various cortical depths as a function of the spin echo EPI echo train length. So here is the cortical depth dependence of the microvascular and PL vein bold signals for a 200 micron PL vein. Now these simulations here kind of match our expectations. On the top panel is a simulation with a short EPI acquisition time, which exhibits minimal large vessel signal for all but the shallowest depths. And on the bottom panel, the simulation for a long EPI acquisition time, where we see this large vessel signal that persists deeper into the cortex. Now, these simulations allow us to calculate a new feature in the cortical depth below which the microvascular signal dominates the bold response, which we call the critical depth. We can calculate this as a function of the spin echo EPI acquisition window or echo training length, and as a function of the presumed size of this PL vessel. So the simulation showed that the microvascular dominance occurs at a depth of about 800 microns, assuming a large 200 micron PL vessel for the longest acquisition time simulated, and this depth becomes steadily smaller with shortened acquisition times as the influence of these large PL vessels was reduced. So note that the simplified model is missing a lot of features, and so this exact value may change as more realism is introduced, and we're currently incorporating these image encoding gradients into our van simulation framework. But this work represents an important milestone for us, I think, in that we're now able to capture the effects of image encoding on the fMRI contrast, and we will be characterizing the effects of image encoding on other forms of fMRI contrast as well. Because, yeah, when we're using gradient echo bold with a gradient echo readout, there's no interaction between contrast encoding and image encoding. But as we move to away from, I should say, gradient echo bold and away from bold more generally, it'll be increasingly important for us to account for how image encoding influences or corrupts the functional contrast. Okay, so the last project I want to present was recent work from Mike Bernier from my group that sought to address large vessel bias in bold fMRI using a novel forward modeling approach, which is really inspired by our van modeling efforts. The motivation here is that if the vascular anatomical data are available, simply masking out bold voxels that large veins omit several features we know that the vessels exhibit. We know that vessel size and orientation relative to B0 influences the extravascular field offsets, as well as the vein contribution also varies with distance from the vessels. And therefore, we should take these features into account when predicting which bold voxels have vascular contamination. For example, a bold voxel next to a vessel that's parallel to B0 does not need any correction. Also, the influence of large veins varies with distance. So even bold voxels that are not immediately next to a vein may exhibit large vessel contamination. And so the goal of this project was to estimate extravascular contributions of the veins measured for each participant that accounts for the geometry of the vessel and the distance from the voxel. And the subject-specific bold forward models based on high-resolution vascular anatomical data. So to compute the vascular segmentation and blood susceptibility that are the basis of this forward model, we estimated blood susceptibility from QSM at 7 Tesla acquired with 3 different head orientations, and used a multi-scale Frangi approach for vascular segmentation on each of these gradient echo acquisitions, and then just combine the results. Now, the 3 orientations were helpful because they allowed us to reduce the number of false positives that occur when the vessel is oriented parallel to the B0 field direction. So the 3 orientations allowed us also to perform quantitative susceptibility mapping, or QSM, and then we're able to combine the QSM maps with the vein segmentation to look only at susceptibility within the vessels. So then with this vascular or blood susceptibility, we computed extravascular field offsets using the same method for our van modeling. the finite perturber method, which is basically reversing the dipole inversion used in QSM. We were then able to compute the extravascular field changes with activation by assuming a simple 10% increase in SO2 above baseline. Okay, so here are the resulting field offsets, which can be seen to exhibit the three properties I noted before: the effective vessel orientation, the effective vessel diameter, and distance from the vessel. So here Mike picked out a few examples where you can see that the field offset is larger for vessels perpendicular to B0 and for vessels with bigger diameters compared to vessels that are parallel to B0. Also note that this map was surprisingly consistent across subjects, which suggests that we may be able to derive an atlas for a simple field offset estimate that does not require high-resolution QSM data in each participant. So how might we use this extravascular field map to interpret or correct our BOLD data? So as in a demonstration, here Mike acquired some very simple 2.5 millimeter BOLD fMRI data in response to visual activation. He then downsampled the field offset map, masked it with the BOLD signal so we can compare the two. This highlights regions with large vein biases. So as a first test, he tried both binary masking with the vascular segmentation shown here on the left, and then normalizing the BOLD activation with the vascular field offset map shown on the right. So as you can see, where there are false positive activations that are displaced away from the visual cortex, these can be identified and removed with this field offset map. Now, this is just an initial test and more validation is needed, plus comparison to other methods, but as a downside, it requires QSM data for each participant. But again, I think that atlas-based approaches, may be an option. Okay, so to recap, I've shown you how we've extended the VAN modeling framework, how we're moving towards a VAN model for humans. We've been incorporating realistic image encoding gradients, and we're also applying a forward modeling approach using the subject-specific vascular maps. Now, so the so-called inverse problem is the term that I've heard several times that refers to relating the measured fMRI signals back to neural activity, similar to methods for deconvolving or calibrating the fMRI data. And we've already heard a bit about this in the first laminar dinner. Here you can see maybe Camille in the corner there in the Zoom screen grab. Also, new work from David Norris's group has demonstrated a new deconvolution approach based on their estimated laminar point spread functions. But I think VAN models can help here by, for example, deriving more realistic laminar point spread functions that are needed for this deconvolution. We can also use them to characterize translaminar flow within the capillary bed, and we're thinking through other ways that I think VANs can help here. So a final question that I wanted to raise about how we should go about localizing neural activity to individual cortical layers. And can we do better than what we're doing today? One approach is model inversion, right? Typically used to correct gradient nickel BOLD, which has the highest sensitivity, but unfortunately the poorest specificity, and depends on many model assumptions. I like to think of this approach as trying to put the genie sort of back in the bottle. Another approach is using more advanced acquisitions, spin echo, non-BOLD, that can achieve better localization, albeit with lower sensitivity. However, in some of these cases, the physiological interpretation is not well understood. Now, both of these approaches have limitations, and while I'm not sure whether today's gradient echo BOLD can really be transformed in a way to provide better specificity than, say, vaso, I do think that this model inversion can help. And I think also that the biophysical modeling approach I presented can be useful to better interpret non-BOLD imaging methods as well. So the difficult part of this modeling is getting the physiology and the vascular hemodynamics correct. So once that's done, the simulations of the MR physics, I think, is more straightforward and a much smaller investment. So I'm not going to try to answer this question now, and I think it'd be fun to discuss with everybody later in the dinner. However, I do think that modeling can be used to improve any form of fMRI. Okay, so just to wrap up, I've shown you how biophysical models can provide some insights into fMRI signal characteristics. They may generalize across different stimulus configurations, cortical depths, and can potentially help evaluate the spatial and temporal resolution limits of the hemodynamics. So VAN models can be used to inform simple, more practical models by providing parameter values or by providing laminar point spread functions for deconvolution. Now, these models are complex, right? But once the relevant parameters are identified, these can help guide experiments to measure the parameter values, generate validation data, and perhaps Evelyn will tell us a little bit more about solutions to acquire these data. Also, these models can help us to build physical intuition, evaluate theories, generate hypotheses that can be tested and hopefully falsified experimentally. Now, so far, these models have mainly been applied to BOLD, but as I mentioned, they can provide insights into more neurally specific fMRI contrasts to further improve their interpretability. So thanks for listening. I'd like to thank my group, past and present, and a few people in particular, Avery, Jorg, Mike, and Grant for contributing slides. I also want to thank Divya, Saskia, and Mukin for helpful discussions about modeling, also useful discussions with Natalia Petradou, Camille Uladag, and Federico Di Martino. And a major thank you to David Boas, Anna DeVore, Louis Gagnon, and David Kleinfeld for helping us to get started on this path. And so one last thing, my lab now has several open positions for postdoctoral fellows to work on data acquisition and modeling and to apply some of these models to interpret our data. And so I'll be posting ads this week, but if anyone here is interested, please feel free to contact me. Yeah, that's it. And with that, I'm looking forward to the discussion. Yeah, thanks everybody.
Very nice. Thank you very much, Jonathan. So because we got started a tiny bit late, I would say maybe we have time for very pressing questions. I see David there. Oh, now I have to figure out how to. Oh, David, right. You're unmuted.
I'm unmuted.
But in the meantime, we'll just start setting up the next talk if you don't mind.
Sorry. Yeah, of course, of course. Hello, David.
Hello, John. Great, really great work. I think you've made a fantastic tool. I was just going to ask you, a lot of the time, we're always worrying about those bold signals adding to each other coherently, and they're not really what we're interested in. And I think you can now look a bit at the time domain, and it's something we've argued several times, is that if you would have a unique variance to a specific layer, it will not add constructively to the variance from other layers. So actually, for what you're really interested in, a signal which is having variance to a specific layer, you don't have to care so much about the signal in those large vessels because it'll be filtered away. But you do need a regressor to identify it, because otherwise it ends up in the noise of your GLM. And I was just going to challenge you maybe to simulate some of these things and put some unique variances in at certain depths and see if you can uniquely get them out again.
No, I think it's a terrific idea, David. And I feel like, yeah, you've shared similar ideas in the past. I mean, I think that you're absolutely right. I mean, the time dimension is something that's currently kind of missing from a lot of our point spread function measurements, first of all. And I think you're absolutely correct. I mean, we have been looking at this a little bit with Daniel Gomez and Laura Lewis to try to understand, how these models can help us to guide experimental design, right? So once we can understand potentially how to drive, specific layers, with specific temporal signatures, and maybe to enhance the specificity this way, I think this can be really valuable. And so, yeah, you're absolutely right. To the point, I didn't have time to go into this, but I think that these models can help a lot with experimental design and, bringing in the time domain as well.
Thanks.